schematic platform for bioinformatics applications, intranet and or internet, clients and servers, browsers, light applications, HTML or XML, Perl or C or C++, Java, BioPerl, MySQL, SQL, bioinformatics tools, statistical analysis are HPC with MPI, www servers, database servers, intensive computing servers, sequence assembly by the shotgun approach, master short sequences simply by examining the sequences for overlaps. No need any prior knowledge of the genome, DNA, 500 base pair or fragments and sequences, overlapped sequences. What is a gene? Where is a gene? The highlighted area is a part of gene. And the next is transcriptional control sites, non-coding RNAs, mRNAs, where nucleotide level annotation. What? Protein level annotation. How? Process level annotation. 3 layers of genome annotation. From Steen L. 2001 Nature Reviews Genetics and evidence-based prediction, cis alignment, trans alignment, ab initio or de novo prediction. Here is the diagram: training dataset, genome annotation flowchart. Training dataset, ab initio only target genome sequence, de novo and low accuracy, high accuracy, and then joint probability models, combiner, and here is the EFL DNAs, cDNAs, ESTS proteins or related genome. Evidence-based aligned DNA sequence from other informant genomes, and then de novo, and then only target genome sequence, and then ab initio. And from if we go from evidence based, then training dataset, and then if you